Okay, so in the second part of this lecture, we're talking about hematopoiesis, is chapter 13. And hemopoiesis and hematopoiesis are two words for the same thing. Points out the continuous replacement of blood. And this process occurs in the bone marrow in skeletal medullary cavities. And that's where you're going to be making all sorts of blood cell types. So we talked a lot about erythrocytes. Erythrocytes are red blood cells. And the process of making more red blood cells would be erythropoiesis. And then there are granulocytes, and we talked about the different types of granulocytes. There are neutrophils, basophils, eosinophils, and the production of new granulocytes is granulopoiesis. And monocytes are a type of agranulocyte, right? There were lymphocytes and monocytes. And monocytes production is called monocytopoiesis. So, I mean, each one of these processes, more platelets, thrombocytopoiesis. So, why are platelets called thrombos? Sometimes because of blood clotting. So, you should, in your mind, you need to start connecting platelet and thrombo, the word thrombolytic, or thrombo meaning a blood clot, because platelets, their role is really to facilitate blood clotting. There's also lymphopoiesis, so that's the other type of agranulocyte, and that you need more of that too. So where does all that stuff come from? And this is one of my favorite subjects, and that is the stem cell. So we've talked about stem cells a little bit before, and there's a lot of interest in biology and in medicine in working with stem cells, and we can manipulate stem cells in the lab and isolate stem cells. And so stem cells are a big deal. So even though in the normal body there are stem cells, and they're not necessarily embryonic stem cells. They don't need to be taken from an embryo or taken from the inner cell mass of an embryo. There are other sources of stem cells that are available in the adult. And so they're in various tissues. And in the blood, there needs to be a lot of stem cells. And the reason is because there's a high turnover of blood cell types. Wherever there's going to be turnover of cells, you're going to have an important role for stem cells. So in the skin, I've spent a lot of time working with skin, and the epithelia of the body keeps being sloughed off, right? It appears as house dust and stuff. I mean, so you wash it off. And so there always needs to be a new epidermis. So that must mean that there are stem cells. And in fact, there are, like every 20 cells or so at the lowest level of the epidermis, that's a stem cell. And it gives rise to daughter cells, sometimes called transit amplifying cells, and those really divide rapidly. But the stem cell population is typically small, and it gives rise, it replenishes itself, so stem cells make new stem cells, but then it also gives rise to these daughter cells. And the daughter cells are the ones that really divide rapidly. Sometimes it's called transit amplifying cell. So one characteristic of a stem cell, though, is that it's pluripotent. And pluripotency means that it could be a variety of different things. So it has that potential. Now, human embryonic stem cells, those are the most pluripotent cells. But some of these other cells in the body are, they could be, sometimes they distinguish multipotent from pluripotent. They might be multipotent cells. Nonetheless, in the literature, they pretty much always talk about pluripotent. Which just means, plural, so multipotent. They can become a lot of different types of cells. And one tool for isolating stem cells is to use a fluorescence-activated cell sorter. And so you need to know that word, FACS. So those fluorescent activated cell sorter. So FACS. And it's a machine, and it typically uses lasers, it uses fluorescent dyes, it makes use of markers. Remember we had talked about markers, there are cell markers. A cell marker is a protein that you have an antibody against, and you can tag it, you can make it glow red or green or something, you know. So it becomes a marker. So the fluorescence activated cell sorter is it's a machine that has a pump, and it pumps your cell solution. You have cells in suspension, and they're pumped through some tubing that goes into the machine. And then there's typically laser light that causes the fluorescence of the marker thing to glow a certain color. And then some of the machines, like an actual sorting machine, will deflect those cells slightly from the other cells. Those are the ones that are, like, let's say, is glowing green because it's got a marker protein that you wanted. It's going to deflect the fluid just slightly, and it will collect as a separate pool from the ones that are not glowing green. So you can select ones that are glowing different colors. Isn't that cool? That's what the whole cell sorting thing is about. So if you use a marker that's unique to stem cells or any other kind of cell marker, you can select populations of cells. So FACS is one way of isolating stem cells. So yeah, so stem cells are really interesting. So we said that there are pools of stem cells in the body, but that's not the same as human embryonic stem cell. HESC, human embryonic stem cell. That's the thing that we're doing research on, and it has the potential to become all sorts of parts of the body and all that. But these are internal stem cells, and the stem cells that appear in the bone marrow are giving rise to blood cell types. And they're able to give rise to all of the blood cell types. So a stem cell in the bone marrow will give rise to everything. It kind of simplifies things. Doesn't it? Isn't that kind of amazing? Like all those, so we talked about the erythrocytes, and we talked about platelets, and we talked about the leukocytes, and there's granular and agranular, and there's lymphocytes and monocytes, and neutrophils and eosinophils, and they're all coming from stem cells. The stem cells are a pluripotent progenitor cell. Well, the progenitor cells are actually a derivative of stem cells, so these are somehow pure. And the next line, I'll get rid of this human embryonic thing. So the stem cells then go to a progenitor cell. So you think of that being the next step in the maturation of a stem cell, is that it becomes some sort of progenitor. And the progenitors have are maybe not so pluripotent, but might be more multipotent. You know, they're sort of funneled towards being a certain thing at that point. And from the progenitor, then you get a bunch of other cells, and these are some sort of blast, like an erythroblast, as opposed to erythrocyte. It's an immature cell. So the progenitors give rise to immature blood cell types. And the progenitors are coming directly from stem cells. So one of the cool things is that in blood, the using cell therapy has been a success. And it's like one of the only areas of success in the whole body using cells. Isn't that amazing? I don't know. I mean, you know, like we want to use the human embryonic stem cells as a therapy. So we're injecting human embryonic stem cells, and sometimes we're injecting progenitor cells, you know, ones that we think might become neurons or whatever. And so we find a location in the spine, and we're trying to cure the person's paralysis by injecting these progenitor cells. But in general, it's been very slow to get terrific results using cell therapy. kind of, it's slow. Slowly happens. But in blood, it's been going on for decades. I mean, we found out that that all the blood cell types are synthesized in bone marrow in the adult, and that if a cancer patient gets irradiated because of a cancer, we use radiation therapy, and so we're trying to kill the rapidly dividing cells, so we use radiation, but then you end up killing the stem cells that are in the bone marrow also, and same thing with chemotherapy sometimes, depending on the compounds. So, you know, sometimes if you damage the stem cells, you could squirt some cells back into the body, and they would go to the right place, and they would replenish the bone marrow and give that person a chance to live. So if somebody goes into Fukushima, I guess is the latest nuclear disasters. You've got some workers at Fukushima, and they go in there to try to fix reactor 3, and there's all this radioactive water and everything. Well, it's going to damage their bone marrow. Some stem cells could be wiped out from that high radiation exposure. And if the guy shows, you know, radiation sickness, you might want to transplant them with some stem cells in the bone marrow specifically that will give rise to new immature blood cell types. So that it works. That's a success. Oops. Okay, so the types of bone marrow. Yeah, and there's this concept of the colony forming unit, CFU. This isn't super important, but the idea, you know, I'm a lab guy, so I mean, whenever I think of cells, cells can grow in a dish, and if you have a cell and it's growing, it's going to give rise to a colony. It's going to divide, and it's going to stay put in its one location. So that becomes a colony. It started out as one cell, but it keeps multiplying. And so these are colonies. They're not cells anymore. They're groups of cells. They're colonies in a dish. So this is a tissue culture dish. So when you think about it in that way, a colony-forming unit is a cell that's going to give rise to a larger colony. And it doesn't have to happen on a dish. It could actually happen inside the bone marrow. And so the different lineages come from clumps of cells, and that's all it means. And so they talked about, you know, the erythroid lineage, the CFUE, and the thrombolytic lineage, which is a megakaryocyte lineage, CFU-meg. You know, but what is this whole CFU thing? Colony forming unit. They're just saying that there's a clump of cells, and that the clump is going to give rise to these blasts. So physically, it's not dispersed; it's clumpy. And so, you know, a clump of progenitor cells are going to give rise to a bunch of blasts. And if all of these blasts are granulocytes, then it's a CFU-GM. For granulocyte monocyte lineage. That's all that means. Okay, and then at the very beginning of this class, I had talked about biotech. And biotech drugs are, you know, now part of the main pharmaceutical industry too. Pharmaceutical companies have bought out a lot of the biotech companies, and they do some of their own biotech type research in-house as well. And so it's really kind of a continuum, but the biotech tends to be kind of newer methods and smaller companies. But when I talked about that, I was mentioning that there's different growth factors. And so there's this idea of a colony stimulating factor. So just to demystify all that, again, we said that the progenitor cells are giving rise to blasts, but they tend to be clumps. And on a tissue culture dish, you can see it. It's like, oh, it's a clump of cells. They're all dividing, and there's a group of them, and then the group gives rise to something. So a colony stimulating factor is a growth factor, and sometimes this is just CSF, colony stimulating factor. It's a growth factor, so you could say growth factor, and that's pretty synonymous with cytokine. Every once in a while there's a nuance, slight difference, but mostly they're kind of synonyms. So it's a type of, growth factors are this big thing, and some of those growth factors are colony stimulating factors. And the colony stimulating factors are the ones that we're talking about right now because they trigger the maturation of a particular cell type within the bone marrow. And so there's, some factors will stimulate more granulocytes, and and we said that erythropoietin would stimulate production of more red blood cells, erythrocytes. Okay, so bone marrow, medullary canals of long bones, it's small cavities of cancellulous bone. There's red bone marrow and yellow bone marrow. The one we're really interested in is red bone marrow because that's where blood forms. The yellow bone marrow is where adipocytes form. So anyway, these factors, that's those are biotech products, and they're things made naturally by your body, and we can modulate blood cell types. So then the bone marrow is separated into red and yellow. And yellow is, for what we're talking about with the blood, is a little bit less interesting because that has more to do with the adipocyte. But it's still super important. Adipocytes are fat cells, but they end up having a lot more biological significance than we've given them credit for, because they're kind of storehouses of some of these factors as well, so it modulates the growth of other things. So when we talk about the bone marrow, what's different about marrow versus blood? Huge difference. Is like, wow, we've waded finally into tissue, actual tissue. This is the beginning of tissue. So here, this is a histology class, and you guys imagine we're going to be looking at slices. Well, from now on, we are going to be looking at slices. Slices, slices, slices the whole time. So, I mean, it's kind of funny that we started out with blood, and we looked at cells in suspension, these little round blobs. But blood is so important because it supplies so much of what tissue needs that it makes sense to talk about blood, I think. But once we get into marrow, we're talking about real tissue, where cells are kind of cemented in with each other, and there's real structure there. So what is the structure? Part of that is stroma. This is sort of a general word. Have you ever seen that word before? Stroma? Stromal. Sometimes they describe something as being a stromal component. Stroma. Gee, it must be Latin, huh? Reticular connective tissue. Stroma, I'm used to thinking of stroma as the support cells. There are some cells that have specific functions, and then there needs to be other cells that are helping to keep those guys alive. And those are stromal cells. Stroma is stromal cells. It's made of stromal cells. And stromal cells tend to be, it's almost equal to fibroblasts, some sort of connective tissue cell type. And this is where, it's so easy to get confused and think biology is endlessly complicated. It's like, well, what are we talking about here? We're just talking about a support cell function. That a fibroblast-like cell is producing a lot of extracellular matrix. And that's sometimes abbreviated ECM, extracellular matrix. Have you ever seen that before? So it's what is that? It's like all those proteins. Collagen is a huge component. You know, collagen 1 is the most common collagen. There's 28, I don't know, something like that. Different types of collagen, and they're all related to each other. Gene coding regions are slightly different, so it adds some additional functionality. But extracellular matrix is made up of just lots of different proteins. Collagen is a real good example. There's a lot of collagen in the body. What's the one most common protein in your body? Hemoglobin? Comes in close. What? Albumin? That's probably a good one too. It's a major blood protein, right? It's in the plasma. Turns out collagen. So, like if you roast some meat and there's, you know, the juice that forms or whatever in that, and then you put it in the refrigerator, it's like you get some fat, but then you got this gelatin stuff. Gelatin is denatured collagen. There's so much collagen in the body that if you cook it, you get gelatin. And then if you add coloring and sugar, you call it Jello. And you put some whipped cream on top, and you get cherry. So collagen is a big deal. It's this triple helical structure. You know, it's part of this extracellular matrix. Other really common proteins, can you think of another common extracellular matrix protein? Fibrin, that's blood. Like fibrinogen goes to fibrin during blood clot. But related to that, close, so it sounds like that. It's a different word, almost the same thing. Fibronectin. Yeah, fibronectin is one. So let's just list a couple of these things. So we have collagen, we have fibronectin, and we're just writing down examples of extracellular matrix. And the matrix, it's like a blanket of threads, threaded proteins, all threaded together, and it forms a matrix. That's why they call it a matrix. I've seen pictures of this too. In the skin, it's really kind of, you know, very visual. If you did an SEM, scanning electron micrograph, and you see these strands, much like the blood clot. You see strands, remember seeing that? And then, you know, they have some different shapes. There's a couple of funny looking things that, you know, kind of pop out at different locations within this whole structure, whatever. But this is not a cell. We're talking structure. We're histologists. We're interested in the structure of the body, of the human body. And now suddenly I'm not talking about cells. I'm not talking about living stuff. I'm talking about proteins. The proteins are made by cells, though. They're made by stromal cells.